of Comparative Biosciences, College of Veterinary Medicine, right?
Mm-hmm.
Yeah. And she's going to be telling us some very interesting things about reproduction rhythms, which Megan knows a lot about. She has worked in this area. I met her at the University of Michigan when she was a postdoc there a few years ago, not so many. But anyway, so I know she's very knowledgeable on the subject. But I was going to give you a little preview of what we're going to do for the next 3 weeks. I think Slim might not need to come anymore. I know it's hard to imagine, Slim. You can listen to what we're going to do and then you can decide. So I've been giving a lot of thought to this, and what I've decided is next week, I told you we were going to have student presentations, but we're going to try to kind of transition into them. So next week, the topic is going to be metabolism. It is huge in rhythms. It's like, it's the pancreas, it's the fat cells, it's the brain. I mean, there are many different aspects of metabolism and rhythms. And so what I'm going to do is send out 1 or 2 review papers, and then I'm going to organize you into groups, and you're going to pick the papers that you present. Your group is. And so, it's going to be just a little step forward from what we've been doing, where we give you the papers and you make the presentations. And then the last week, which is 4 weeks from today, but 2 weeks after that day, you're going to pick your own topics as groups and make group presentations on the topic that you choose. And there are things we didn't cover. There are other things to cover that we've done. But so then there will be a component of you guys coming up with an idea. 2 weeks from today, I decided we're going to do a writing exercise, and we did this in my lab group meeting a couple years ago, and everybody doesn't have to chime in and say how they liked it, but my sense was they liked it a lot. And that is, we're going to have an exercise on writing specific aims for a grant. And this is something that you do all the way through your graduate training, you do it all the way through if you become a research scientist or any kind, if you ever want grant money, you have to do it. Notice Megan and I were talking about grants shortly ago.
That's why we look sad.
But it's going to be just a 1-page essay in the format that's a very standard format. And I have this book. You should probably need this book, Megan.
I probably have 3 copies of it.
It's about grantsmanship, and I'm going to send out the section on how to write specific aims. The specific aim page is 1 page long for any grant, no matter what size it is. And it has the general background, usually, and I'm going to give a little talk on this next week. The first line is, what would you tell your congressman if you were on the elevator for 3 minutes about why you should get funded? And this is where we all go wrong, all of us scientists, because we're all so into, the depths of what the details. You can't give people the big picture and, why taxpayer money should go into supporting your research. So that's very important to do. So there's that paragraph, and then there's a paragraph that brings you more specifically into the research area you're in, what we don't know, what we'd like to know. And you won't know all this, but this is just what you have to think about. And then you have your aims, and then, what's the why would the world care? How many people is the NIH going to be able to think about helping if this research is funded? So that's going to be the exercise from 2 weeks from today. And I don't think any of the people from my lab who are in here have done this so recently that it won't be new to them. But the idea is for you to pick anything that we've talked about, because that'll make it easier for you because you've already heard it introduced. I know this is possible because I asked Lucas, because his curmudgeon TA organizer kept making him be a proctor during class, and the third time this happened, I said, okay, Lucas, it's not enough just to summarize the paper. I want you to tell me about an experiment you do. He did a beautiful job. You did a really nice job in that paragraph. And it's that kind of thing. Just think of an area you'd like to know more about and 3 kinds of ways you could address it. And I think it'll be an interesting way to build on what we've learned. It'll be an interesting way for you to learn about grants. And it'll be a little experience in writing. And what I can also tell you, I have written many, many grants. I have received a number of grants, but I've written many, many more that I've been funded for. And what I can tell you is simpler is better. Simpler writing is always better if you're trying to communicate an idea to a committee who doesn't want to read your paper or your grant more than once. And so that's another part of the exercise, is making it simple and accessible. So this is just a preview to what I'm going to tell you more about next week. But that's what's going to be coming, and I'll be sending out on Friday the information about the groups and, you know, the paper on metabolism, and then you can talk to email your group and pick some area of metabolism. That's a wonderful topic. So anyway, with no further ado, Megan Mahoney is going to make a presentation today.
Does this control slides?
I don't know.
I don't
think
either.
so.
I don't think so either. That cramps my style. I'm kind of a spaz.
I know.
You know.
I know what you mean.
Okay, so as Martha said, this is what I do. I study reproduction and circadian rhythms and their interactions. So today, if I have time to get through it, we're going to briefly touch on how reproduction is organized by the hypothalamus pituitary gonadal axis. We'll talk about how we know that rhythms regulates reproductive events in rodents. We'll talk about the reverse, how we know hormones regulates the expression of rhythms. And then we'll talk a little bit about humans and what we know in humans, trying to parallel those results to what we know in rodent work. And then at the end, talk about shift work. Okay, so for a brief summary then of how reproduction in the female, and that's mostly what I focus on females, but that's mostly what this talk is focusing on. It's regulated by cells in the hypothalamus, GnRH cells, who release their product that comes down and acts on the pituitary. The pituitary, stimulated by the GnRH, secretes luteinizing hormone, LH, as well as FSH. And then at the time when LH gets released, it's usually released in a pulsatile manner. In rodents, every 4 days, there's something called an LH surge. That circulates in the blood to get to the ovary, where it causes the release of the egg. The follicle ruptures and ovulation occurs. Okay? So there is feedback where we've got the ovary develops and makes increasing levels of estrogen. Okay, so hypothalamus pituitary gonads, we have this estradiol that feeds back and talks to the hypothalamus. Okay, the GnRH neurons are getting this hormone signal and deciding when they're going to release their product. Okay, and again, they cause the release of luteinizing hormone. So I'm going to tell you that in addition to needing GnRH neurons and estrogen levels in order to induce ovulation, in rodents we have a lot of data that the circadian clock is involved in this. Okay, so you can map out all the hormones that are involved, and these hormone changes that I'm going to show you here are the same as what would occur in the human menstrual or reproductive cycle. Okay, but it just happens on a larger time scale. So here we have increasing levels of estrogen as the follicle grows. More and more estrogen happens. This is, what have they done? 1, 2, 3. This is a 5-day estrous cycle. We call it estrous cycle in rodents. We'll call it reproductive cycle in humans. Estrogen increases at some point, and then we have LH. We'll have a big surge. Okay, and after LH, sorry, I got to back up. Estrogen causes this change in GnRH, which causes this surge of LH, okay? And then ovulation happens. Following all that, progesterone goes up. Okay, so now notice that we're talking about circadian rhythms, and this is, I said 5 days, and this is only happening once every 5 days. Well, 4 or 5 days. So how can I tell you that there's a circadian rhythm to all of this if this is happening every 4 or 5 days? By the way, you can divide up the estrous cycle into different stages. They do this based both on the hormonal states and cytology, vaginal cytology. So diestrus, proestrus, estrus, and medestrus. Okay? So here's all these events that I've just told you about with the hormonal changes. And here is our estradiol going up, increasing, and with this resulting surge in LH. All right. Why does it only happen every 4 days? How could you even get a 4-day cycle like that, circadian cycle? All right, so if you ovaryectomize a female rodent and give back estrogen, so here this little red dashed line is mimicking if I gave them a constant source of estrogen. Okay? If I gave them that constant source, you end up having a daily LH surge. Okay? So there is definitely a circadian timing signal that comes every day and tells the animal when to have an LH surge at a certain time of day. Now I want to also point out that it's definitely only happening at one time a day. despite having this chronically elevated levels of estrogen. It's not like the animal just freaks out and dumps all of its LH all at once. There's a window of time when that LH is getting secreted. Okay? So we do know then that there is a circadian timing signal. We know that these cycle lengths are very regular. In the rodents, it's either 4 or 5 days. It's not 4.5. Okay, you can't really have a half day. All right, so that's LH surge. I'll get to the SCN in a minute. I'm assuming you'll know that. Why I left that little slide like, hey, everybody, look at these rodents having sex. Okay, so another reproductive event controlled by the HPG axis would be mating. Okay, so I'm going to assume you all know actograms. Yes? Yeah, you're like, enough of the actograms. Okay, so this is a female rodent, and she's free running. And so, of course, these are all erectivity things. Let's see if I put the dots on this one. Okay, so they've put little dots on this figure for the days when that female went into heat, that day of the estrous cycle that we call estrus. Okay, you may actually notice, since this is a hamster, you may actually notice that on that day, when they've put a dot, she actually had a phase advance. She woke up earlier. And in general, the activity is, the alpha is elongated on that day when she's in heat, when estradiol is highest. Okay? So we have these dots indicating that every 4 days, this hamster is having, going into heat. All right? So an additional piece of evidence then that this is run by the circadian system is that the estrous cycle free runs. Okay? And instead of free running, it free runs, what do they call a quadridian rhythm, okay? So the animal is having an estrus cycle an estrus and an LH surge every 4 days. And it's 4 times the tau of that animal, if that makes sense. So if the animal's free running period is 23.2, 4 times 23.2 hours, every unit that's about that, and so it can be 95.36 to 97.54 in this paper. That's when that animal has that LH surge. Okay? Further evidence then, so this happens to be an LL. We have this LH surge happens and then the activity happens. So the LH surge happens right before activity onset. And it's phase locked. The relationship of that is always the same. So it's between 1 and 4 hours, but this is a really tight relationship. Okay, you phase advance activity, you're phase advancing the LH surge. Okay, the other thing that's happening in this, I guess I'll go back to this one, is that they gave this animal something that we call heavy water. It's radioactive, I think. I haven't thought about this paper in a while, but and it lengthens the tau. Okay, and so the same thing happens. This animal gets a longer tau, and that estrus cycle free runs, matching the length of that new tau. Okay? All right. So here's another reproductive event that happens, and this would be the timing of birth or parturition. Animals, of course, it would make sense that you wouldn't want to be like out and eating or running from a predator and then suddenly you're giving birth in the middle of all that. That kind of would be poor planning. So it would be best to be timing your birth that occurs when you're in your little nest, okay? So in this paper, I liked how they named different countries here. They ended up phase advancing and delaying these animals at different times to try and see if the animals were going to give birth with a duration of time, or if they were going to time it to a light-dark cycle. And you can see, here's our control animals. They clearly are giving birth during the lights-on period. These are rats. They don't want to give birth when they're active, as we mentioned. And another interesting thing to this pattern is, so the majority of them are happening here in the light. A few animals gave birth during the dark, and then there's another kind of smaller clump over here. They did do some delaying and advancing of animals. These phase-advanced animals were able to keep their birth happening during the time of lights on. These delaying animals had a little bit of a problem. You can see some that ended up giving birth during their active period, or at least during the dark. But for the most part, these animals are basically entraining their rhythm so that way they can give birth during their time of inactivity. Okay? So we have all these reproductive events. And they all appear to be circadian, but people have gone on to establish that the SCN is critical to these events. So here is a hamster. You can see they've got our activity double plotted. And here they put 2 little pluses for each of those days that the animal was in estrus. People love hamsters for reproduction and circadian biology because they're so precise, and their estrus cycles happen every 4 days. Okay. This animal stopped having estrus cycles after the SCN lesion, and then they free ran it, and those estrus cycles never came back. As a side note, if you plant the SCN back in, diffusible factors, do you guys hear about those? You never get estrus cycles back. You get activity patterns back, but not estrus, indicating that the estrus rhythmicity regulation needs a neural signal and a neural connection, and not just a diffusible factor. Okay. All right. People have taken this farther, and if you take hamsters and put them into constant light, you can get them to split their activity bout into 2 components. All right, a morning bout and an evening bout. In a female hamster, you can get these 2 components to each have an LH surge phase locked to the onset of activity of each of those bouts. Okay, so here they put it in red and blue. Sorry, colorblind people. And they've put a little sign like, so here that star would be where this LH surge happened, and that star is where that one happened. So they end up having the phase locked relationship that we've already talked about. And but they have 2 LH surges essentially. Okay. Also interesting is that LH surge happens to be about half the amplitude of a full LH surge. Okay.
Physiologically, what does that do?
Whether or not they really ovulate?
Pregnant at either time? If they made it at either time?
I suppose they would.
You know, what's the consequence of that?
I don't think anyone's ever followed up on it.
No.
I bet, you know what, I bet they have. I bet they just haven't published it.
I think Lucas had a question over there.
Yeah, so
Yeah.
you added the deuterium water.
Yeah.
So what was the physiological significance of that?
It ends up lengthening the tau, the free running clock, but I'm not sure what the mechanism is underlying that. Do you know?
You know, people, you irradiate animals, you
Yeah.
change the period. Nobody knows. It's sort of like, people used to do those kind of manipulations. Sort of gee whiz, whether you're changing the, mutating clock genes. Could be something like that, but nobody knows for sure.
It changes it consistently?
This animal actually looks like it affected it for a period of time and then it regained its original tau. Just because I see this angle and that angle the same. It lengthened it for a while.
Nobody really knows.
Yeah. That's probably from
Those are
the
like old
70s or something.
experiments, and they're still out there.
Yeah.
Why did they try them?
Yeah. Yeah, there's not a whole lot of research on females and reproduction, so even though I'm showing you stuff from 1976, I still cite this stuff. There's not much. Well, there's getting more new stuff. Okay, so here's our split female hamster. She's got 2 different activity bouts. She's got 2 LH surges. Then they did this really cool work, okay? So I've mentioned already we had the hypothalamus pituitary gonadal axis, and so we've got G and H neurons that we know need to trigger that LH surge. So they looked in the brains of these animals. They killed animals at each of these LH surges, and they stained them for GnRH and FOS. And what they found was a hemi-SCN lit up with FOS. Remember, they're having their LH surge right before lights off, right before they're active. So you're going to expect to see FOS because it's the light might have stimulated it. And so they have more FOS on one side. It literally, if I have a picture, I'll have to show you a picture. It's completely filled with FOS on one side and completely blank on the other. And correspondingly, this picture here and this one here are showing you a cross-section through the brain. This is a little rostral to where the SCN is. This is an optic chiasm in the 3rd ventricle. These are where all these G and H neurons are. I think they're really super beautiful, and I would get one as a tattoo if I could. After I get hormones tattooed on my fingers. Hormones. Okay, I digress. All right. So these G and H neurons are these huge bipolar neurons, and they have long dendritic processes and axons. And so here they're indicating that this G and H neuron is active. At the time of the LH surge, you can stain this tissue for FOS in the nucleus of G and H neurons. About 40% of them will express that, indicating that they were transcriptionally busy. Okay? G and H neurons, only 1/2 of that brain will express FOS. Okay? And it happens to correspond to that same half of the brain where the SCN lit up. And if you kill animals at this one, they also have a hemi-SCN and G and H neurons that match the same side of the brain. Okay. I can't tell you like it's always going to be left, right, left, right. I mean, that's the assumption because clearly this animal already died and we can't check it at a later time point. Okay. But that's the presumption. Okay. So evidence then that our circadian system is regulating reproductive events, and this is an incomplete list. I could go on and on and on. We know there's a daily rhythm in the timing of the LH surge. We actually know there's a daily rhythm in mating as well. We know that the LH surge and estrus, that period of heat, free runs with a period 4 times tau, and we can phase shift it, and when they phase shift, they remain phase locked to one another. Then we know lesions of the SCN eliminates our estrus cyclicity, it never comes back. And split hamsters indicate that the SCN is transcriptionally active at the time of the LH surge. So we get a pretty good idea about that the circadian system is critical, and I'm going to say specifically in rodents, for timing these reproductive events. Okay. So what about the reverse relationship? Do reproductive hormones alter rhythms? And the answer is yes. We know in females, I've already just pointed out that every 4 days a hamster becomes more active and she runs a lot more in her wheel. So we know that there are changes across the estrous cycle. When hormones are changing, we can look at activity in gonad-ectomized animals compared to intact ones, both males and females. And we know hormones change during pregnancy. We also see a change in rhythms. We know that it changes just activity in a light-dark cycle. We know gonadal hormones modifies our free-running tau. And there's a lot of new evidence, it's not a lot, but there's new evidence saying that there are hormone receptors within the SCN that could be responsible for how hormones feeds back onto the circadian clock. All right. Okay, so hormones. If female hamsters that are intact, female hamsters, rats, dagoos, surprisingly not mice, bummer for me. Rats, grass rats, whatever female rodent you can think of, except for the one I work on apparently, which stinks. When estrogen is high, they're going to run more, they're going to have a phase advance, and they're going to have a huge amount of activity. I like to think of it as that they're up and running in their wheel because they're looking for a mate. I got to get out there. I'm only in heat for 4 hours, okay? We also know that their free running period gets shortened on that day. If I replace estrogen back to an ovexed animal, ovary-ectomized animal, the free running period will be shorter. Female hamsters have an interesting pattern of activity onset called scalloping. And I was trying to allude to that. I think this is a Jen Swan paper where she had this. So they call it scalloping because it kind of looks like a little, she wakes up early on her estrus day, and then actually the next day she has a phase delay. I like to think she has to sleep in because she's so tired of all that running she did the day before. So they refer to this as scalloping. It's really obvious in hamsters and less obvious in other animals. Okay, but so they know from these intact studies and again from the OVEX studies that if you give back estrogen or estrogen is high, then the tau is shorter. Okay. All right. We know that progesterone, which is the hormone that comes on board after ovulation, also lengthens circadian rhythms. It always seems to, in general, do the opposite of what estrogen does. All right. But we also know that there are sex steroid hormones that affect daily activity rhythms in males. So here's my small shout out to boys. Okay. All right. So here's a couple of examples. So we have 2 different individual animals, and you can see them free running in constant darkness up there. And so the int is intact, and GDX is after they removed their gonads. You can see an incredible difference in the amount of activity as soon as you remove their major source of androgens. Okay. You can also start mapping it, following their gonadectomy. It actually rearranges when they do want to be awake and running versus asleep. So it changes that relationship as well. So the alpha changes as well as the free running period and the level of activity. How do they know which hormone was responsible for this? They took out the gonads. Well, androgen receptors accept both testosterone and dihydrotestosterone. They're 2 separate androgens. And so here is testosterone propionate. They replaced it to a gonad-ectomized animal. And here's an animal that got DHT, another androgen. So you can see very little activity, and replacing the hormone, they get a restoration of activity.
The hydrotestosterone is not that convincing. I mean, it's restored, but look at the difference.
And that's not the best picture because they actually really believe the whole thing is an androgen effect. Other papers from Ray Silver's lab all say that it's just pure androgen. I think she'd accept it if I talked with her and said, I think there's an estrogen effect, but she's sold on the androgen. Oh, yeah, because the other one is if you give all these androgens back to females, you get the exact same results. Okay, so here's period, and they're showing intact animals, animals that are castrated, and then animals getting back androgens, and these two different kinds, and you can see the restoration of it. This little black dot is because they're indicating that there's a sex difference in period. Here's activity duration, alpha, and amount of activity. And again, as soon as you castrate these animals, their activity goes down, their alpha, their whole period when they feel like running gets a lot shorter, and hormone replacement brings that back. Yeah, but you're right over there. That one didn't seem to bring back the amount as much. But I think in other papers it does. Okay, so they give back all these hormones. And so in males, the effect is the same as what I've been describing for females. All right. Okay, so what about these mechanisms? Well, in this male work, they looked at androgen receptors, and it turns out the SCN is chock full of androgen receptors. And boy mice have more than girl mice. If you castrate these animals, you eliminate those androgen receptors. And if you give them back hormone, you can restore those sex differences. You can amp it back up. Okay. And as I mentioned, the gonadectomizing, the males alters their pattern of activity. You lose some lot of it. And you can restore it. Let's see if I can picture. Okay. So here's male and female, and that top picture is just to orient you to the SCN. So they stained that for arginine vasopressin. This one, which is kind of dim, is androgen receptors, and then they show you the overlay. So boy mice have a ton of androgen receptors that females don't have. Okay. See, I'm all about the estrogen receptors, so there's estrogen receptors there too, just not a lot. Okay. Oh my goodness, I spelled rhythms wrong. Look at that. Shame on me. Wow. I'm going to be drummed out of SRBR. Okay. All right, human rhythms. Is there a daily rhythm in these same reproductive events that I've just told you about in humans? What about LH and ovulation? What about human mating behavior? What about the timing of birth? So let's talk about those in women. The end-all, be-all kind of bottom line for human work in reproduction and circadian rhythms is that circadian rhythms may not be nearly as important to, I should say animals, because it's true of women as well as other species that have this long estrous cycle as they are to animals that have a short one, the 4-day one, okay? So humans, again, have that same hormonal events that I was telling you about in rodents, but the days are extended, so it's a lot longer. We still have that rise in estrogen, just takes a long period of time. We still have an LH surge. And then after the follicle ruptures and releases the egg, we have a corpus luteum that forms. Okay, so that follicle kind of scrunches up and becomes a new structure. The corpus luteum is making progesterone. The follicle is what makes estrogen. Okay, so we can actually call that a follicular phase, and we call this one a luteal phase for that reason. Okay, most rodents don't have a true luteal phase. The progesterone goes up, but not for very long. Okay. Okay. All right. She means business. Okay. So are there rhythms associated with the menstrual cycle the same way rhythms are associated with changes in the estrous cycle? Yeah, this is an example of risk-taking behavior changing at different stages of the menstrual cycle. Menstruation, post-menstruation, ovulation, premenstrual stages. Okay. Changes in food craving, changes in mood. Women are more likely to have depressive symptoms during the luteal phase of their cycle than in the follicular phase. Okay. In fact, there's even the premenstrual dysphoria disorder where there's a huge change in mood that's linked to the menstrual cycle stages. Food cravings. I think you might get anecdotes from your friends about changes in activity levels, etc. Okay.
What is the pill, non-pill?
Oh, it's contraception and non-contraception. So cycling women are in white. Okay. So risk-taking scores. At the time of ovulation, it's it's lowest at ovulation, but she's having a normal cycle, these ones in white bars. And the ones that are on the pill have their reproductive hormones kind of clamped with contraception. Okay, so is there circadian rhythm in the timing of the LH surge? Now, I said the LH surge is phase locked to the timing of activity. Humans are day active or diurnal. They wake up during the day. So is the LH surge happening when they wake up? So here's 8 a.m. to noon. Here's 4 to 8 a.m. This was a study using 155 women. And they did get the majority. This is frequency of the LH surge, so the number of individuals. And they do get the majority of them having an LH surge at these two times. I mean, it's a big window, midnight to 8 a.m., okay? A later study with a smaller number of women, it was only about 16, actually narrowed it down and said it happened between 4 and 8 a.m., okay? Great, so circadian system is important, right? Not really, actually, okay? So in this study, they brought in intact, I shouldn't call women intact, women with their ovaries came on in. And they did a continuous constant routine, so they kept the women up for 32 hours, so that's why their activity pattern up there is constant. The gray period was when they normally wanted to sleep. Body temperature maintained a rhythmicity where it went down at times when they normally would be sleeping. I mentioned LH can be pulsatile, and so all those little triangles are indicating when it went up, which is kind of an hourly thing. But they did not get a rhythm. So this is one individual woman, okay? They did not get a rhythm in LH or FSH. Okay, so this is in the early follicular phase, so not at the time of the LH surge, but they're not getting a daily rhythm in this. Here's all the women they had, so they have 11. This is between days 1 and 7. Like I said, early follicular. Absolutely no circadian rhythm. And their definition of no circadian rhythm was that they fit lines to it, and so they get a sinusoidal curve is the best one for temperature, and linears were the best ones for LH. Okay, so does that mean the circadian system is not important for the LH surge? Well, all the grant reviewers are convinced that. I do not need to study it because of that. Oh, I'm getting bitter. Okay. All right. All right, what about timing of mating? In rodents, again, timing of mating happens on that one day when estrogen is high, and it is phase locked. You can't make a mate at all times. They're only going to mate at one window. Humans, with our estrous reproductive cycles, we have a menstrual cycle, not an estrous cycle. Humans and primates can copulate at any stage across the cycle, okay? So it doesn't matter if estrogen's high or not. So we have this unique ability to not have that event timed to hormones. There is a strong circadian component to intercourse with a major peak at late night and a small peak in the early morning. So really the question is that circadian? What do you think? No one's going to be like, I'll tell you when I have intercourse and I'll tell you if it's circadian. Okay, you're not going to tell me. Fine. Fine. All right. Humans do have a pattern at mating, and it does actually correspond to estrogen secretion, but does it have a circadian component? So here's time of day, and here's where people are having intercourse. They're doing it right before sleep. This happens to be college-age people in this study. Yeah, they don't ask people with kids, that's for sure. Okay, so they did actually ask them. No, really, it's factual, not personal, okay? All right. Don't be like, now I know too much about her. No, I didn't tell you anything about me, trust me. Okay, so they did interview these people or ask them in a survey about why they were having intercourse at these times of day, and it comes out to societal or personal issues like, that's when we were in bed, or that's when I wasn't tired, right? So it's very unlikely that this rhythm is a circadian rhythm in humans. All right. There is a rhythm in human mating linked to estrogen, though. So here's females that are initiating. So female initiated is here in black bars. And so females at the time they're ovulating, estrogen's high, they're more likely to initiate than at other stages. And this is where they point out that if you just looked at couples and how often they had intercourse, then they'd have this line. But if you looked at when women were initiating, if you were going to ask that question, then the pattern, a different pattern emerges. Okay. Okay, so that's, and of course, what about timing of birth? Well, you all probably know someone who was in labor for days and days and days, but they can look at labor onset. I said in rodents they're going to give birth when they're in their burrow. We're diurnal, so we're going to give birth when we are inactive, so typically at night. There's been a gajillion studies, and they all typically find the same thing, although sometimes the timing shifts a little bit. So here is labor onset and rupture of membranes, and they looked at the time of when those things happen. One study looked at when women showed up at the hospital, which I think may not have been nearly as accurate. But at least they can say labor begins at times of rest, as occurs in other animals. So you see this big peak here, kind of around midnight. Okay. All right, so I could show you a gajillion pictures of that. Okay, so humans may or may not be circadian. I think the jury's out on that. What if we asked what happens if you disrupt rhythms? What if we changed rhythms in humans and then saw if that impacted reproduction? So this is a International Agency for Cancer. This came out in 2010. This was their top 20 list of things that caused cancer. Shift work is number 19, okay? So everything else on this is a tangible, I'm a chemical, I'm a fume, okay? And then we have this one kind of ephemeral thing, you can't really put it in a jar, etc. And it's shift work. So they say shift work is a significant cancer-causing agent, okay? And we all know, we've all experienced jet lag. Probably we haven't done shift work as much. Vacationers, sleep-deprived students, yes, you really are phase shifting yourself. Traveling athletes, they have sleep disruptions. You get sleepy at the wrong times of day. You're wide awake at other times of day. There's definitely memory impairments. Gastrointestinal upset is one of the most significant ones, changes in appetite. Okay, so this would be like an acute change in rhythms. All right, what happens to female fertility and reproductive cycles? So that's kind of the questions I like to ask. All right, so female shift workers and airline crew. So I just point out that this is now kind of more chronic. They're more likely to have irregular menstrual cycles, either lengthening or shortening. They're more likely to experience pain during their menstrual cycle. They have a much harder time getting pregnant. Once they get pregnant, they're more likely to miscarry or have preterm birth. When their babies are born, their babies are typically more likely to have low birth weight. Women who are shift working do have an increased risk of breast cancer. And if women are going to have a C-section, they've looked at medical outcomes of C-sections given during the morning and the afternoon and at night. If they have it at night, there's an increase in medical complications. I hesitated putting that last one on because it was things like infection, and I thought maybe the problem was the doctors who were sleep deprived and not the woman. So I'm not sure what shift work issue that was. Okay. Again, though, the jury's kind of out on this. So not all studies find this significant effective shift work. Okay. Sure, the risk of breast cancer could be there, but typically most studies find it it's only moderate, and most times they only find it in women working on shifts for 15 years or 20 years. Some studies have not found any impact of shift work on menstrual cycle regularity. Shift work has a minimal impact on preterm birth and birth weight, so people aren't finding a lot of effect here. And shift work doesn't really seem to impact other issues like gestational hypertension or preeclampsia, so it's not increasing those problems. All right, so I tell you all this stuff about rodents and then say this is really significant, and then you go to the human literature and it's tougher sometimes to find a significant health problem. Now you can come up with lots of reasons why. Some of it can be that people self-select right out of shift work, so people who don't handle it as well can choose not to do it. And people who stick around for 20 years stay with it longer, okay? So that's one issue. Yeah, that's one of the bigger ones that comes up, okay. We can look at disruptions in rhythms by looking at clock knockout animals. And here, again, we begin to see all those problems, okay? So first of all, clock gene rhythms, even if they're slave oscillators, are found in the ovary, the uterus, the fallopian tubes, every reproductive organ you can think of, basically. We know in PER1 and PER2 knockout mice, KO mice, they typically have irregular estrous cycles and decreased fertility. So they are still having hormonal changes, but it's just not working as well. Clock knockout mice, this was one of the first studies that really looked at all of the different ones, the different issues that could happen. They had irregular estrous cycles. Their LH surge just wasn't happening as much. They had decreased fertility, and they think with the knockout of that gene, they had trouble giving birth, like it was missing something. B-mall knockout mice, they mate, but they don't produce young. And you may not even see that published too much, because my friend who did that one said she couldn't get them to have sex at all. So they just weren't into it. Yeah, she said they were weak little things.
So do they get them all from heterozygotes?
They must, yeah. That was Takahashi had those. That was, or Maneker. It was Jen Mohawk who was doing it. She said they just were tiny little sick things that didn't feel like doing it. All right. So what about clock genes in women? Do we know anything about clock genes in people? Good question. So, of course, we can't go mutate clock genes, but they can find clock gene polymorphisms, slight variations in the sequence for each of these genes. And so they can look at NPAS2 and clock variants. So, and they do see certain variants associated with an increased risk of breast cancer. They do see polymorphisms in the aryl hydrocarbon receptor, nuclear translocator, blah, 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 something like that, ARNL. Are related to an increase in pregnancies and miscarriages. Women with this certain ARNL1 had both an increase in the number of pregnancies and an increase in the number of miscarriages. Okay. And women with this NPAS2 variant had a decrease in the number of pregnancies. So there is some link of clock genes to reproductive outcomes varying in people. But again, these, of course, are all correlational studies. Okay. All right, so that's what I want to tell you, because I knew your class was probably an hour. Do you have any questions? I do talk a mile a minute. I know I do. I can show you. I'm going to show you another funny one. Okay. This is another human one, 207,000. This is the one where women present to the hospital with uterine contractions. Oh, I want to show you. There's another funny one about human rhythms. Oh, here. Whoa! Frickin' MacBook. All right. I'm not a Mac person. Okay. Okay. That's the rhythm and when people have sex. And so it's a weekly rhythm. Okay? So they looked at random distribution if you're going to do it every day, and that would be your average, but really it's just on Saturday. So it's probably a, yeah. Okay. Okay, so I really could go on and on about all sorts of clock genes and rhythms, but I will leave it at that. Because I've been told I can overwhelm people with too much information. I do this to undergrads when I'm like interviewing them. I'm like, let me tell you what I do. And then I do that to them. Yeah. All right. Yeah.
Do hormones like luteinizing hormone and follicle stimulating hormone, do they have, well, I guess they're under control of the hypothalamus, but does GnRH, does that have like EBOX elements upstream of it, or?
GnRH neurons within the brain, oh no, that's estrogen receptors. GnRH neurons have clock genes within them. I think they find it more likely with it when they do it in the dish more than in vivo tissue, but they do find that. Luteinizing hormone and follicle stimulating hormone do synchronize clock genes on the ovary. So if you put ovarian tissue in a dish and it was cycling with PER genes, if you dumped those hormones on it, seems to be important. So in one study, they took the ovaries of rats that had not had babies or even had estrous cycles, and they put them in a dish, and there was no clock gene rhythms in these immature ovaries. They dumped LH on it, and PER rhythm started. So there is a possibility that the LH is some kind of factor to start that. Yeah.
So if you like castrate the animal or like over sectomize the animal, does the receptor in the SCN for these sex hormones go away?
Yeah, well, yeah, I'm not sure if anyone's looked at estrogen receptors. For the boy mice, yes, it definitely goes away.
But if you put it back, the testosterone comes back.
Yep, if you put back testosterone on either male or female mice, you will get androgen receptors in both sexes. But females ordinarily don't have androgen receptors.
So it's just getting stimulated and you're presenting with the receptors.
Yeah. So in that story about hormones acting on receptors, I like to think about estrogen and its effects on circadian rhythm because I think about females. In general, no one's really looked at that. The consensus would be, though, that if estrogen increases activity, it's doing it by acting on estrogen receptors in the medial preoptic area outside of the SCN, primarily because there's only about, of all the SCN cells, only like 4% of them have estrogen receptors. And there's more estrogen receptor beta than alpha. So that's why the people who do the androgen receptor story believe androgen receptor is really much more important because it's everywhere. And how could such a few small number of estrogen receptors have an impact? But it's a good question about what happens to estrogen receptors in the SCN in ovexed females. We know that estrogen feeding back changes per genes, both in the brain and in the ovary. So in female rats, we've seen that. But again, no one's thought of that mechanism about whether it has to be hormones directly on the SCN or if it could just be hormones that talk to the POA that talks to the SCN.
So would you therefore predict that whatever is happening on the day where they start running early and have the long active period, that's not acting at the SCN, but it's some other site that
That's the
initiates?
thought. There's also the possibility that if estrogen increases activity, people have even talked about it being like a peripheral activation, it just makes them run more jittery or something. And then you get the additional issue of increased activity has a non-photic feedback effect on the clock, and there's a direct link to the amount of activity and tau. Tau is shorter in animals that run more and have a longer alpha. So you have to separate some of those things out as well. No one's looked at hormones and their effect developmentally, so I like to think about that issue as well. I mean, you may have heard about developmental issues with respect to circadian rhythms, but we certainly, alcoholism, there's one guy who did a bunch of alcohol effects on the SCN, but no one's looked at hormones, and yet we're really concerned about BPA and all sorts of other environmental estrogens, so I think that's something to think about. And that doesn't need estrogen receptors in the SCN or anywhere else to have those changes. Yeah, but no, the general consensus is that the estrogen receptors in the SCN are not needed. And I may be the only one who cares, so I may go figure that out. Yeah. Yeah.
This is kind of a question about the diffusible factors. So with a SCN ablation, we lose all hormone cycles. Do you think that, or do you know if the GnRH cells in I believe it's the arcuate, do they express VIP or AVP receptors?
No,
No,
kispeptin's what you're thinking of that's in the arcuate, and GnRH is all in the preoptic area.
Oh, okay.
GnRH neurons, probably I should publish this, have VIP receptor 2 on them. That changes with changes in the estrous cycle. I should really get on that. That was my hypothesis for the timing of the LH surge, that the increasing estrogen is telling GnRH neurons what time of day it is, or basically you should get busy, and that's when the VIP receptors show up. So the daily signal's coming, why isn't anything happening? And it only happens every 4 days. It's got to coincide with that high estrogen. So yeah, they do have VIP receptors. There's a sex difference, if I remember, in VIP receptors on GnRH neurons, and with boys having more than girls. There's a couple papers about that. Yeah. Well, thank you. Thank you for your computer and your adapter.